Hi everyone, this is Mr. Sinti, and today I'd like to talk to you about more of the interesting stories that led us to our understanding the DNA as the genetic material. What's interesting is from our previous talk we were discussing the role that bacteria played in the search for the genetic material. We talked about how bacteria were being transformed by other bacteria, and Dr. Griffith was perplexed by that, and then Avery and colleagues sort of isolated the material that caused the transformation of bacteria, and it turned out to be DNA. But it wasn't enough, because you remember scientists were still skeptical about what molecule was in fact the genetic material, and it really comes down to two molecules in particular. It comes down to, is DNA the genetic material? Yes. Or is it protein the genetic material? And today's conversation is about, again, another classic in biology, and it was performed by Hershey and Chase. And what's interesting about this particular experiment is it was the home run, it was the clincher, it was the walk-off experiment that conclusively showed that DNA is the genetic material. And what's fascinating about this is that it also comes from experiments working with microorganisms. And in this case, we're working with viruses that infect bacteria. And by studying that, Hershey and Chase determined that DNA was the genetic material. And so let's get into that conversation. And so a little background is needed to understand this. And so first thing I want to say is that viruses, as you can see here, let me make that a little bit larger. Let's see if we can go take it up a notch. Let's go here like that. And let's click this. First thing I want to say is that this is a picture of a virus, and this picture is not something that you could see with the light microscope, but this is a transmission electron microscope. This is one of our most powerful microscopes. And so let me just sort of point out a few features of a virus you may not be familiar with. So a virus, first off, are you ready for this? A virus is not classified as a living thing. So it's not a cell, it's not alive, but it's considered to be a biological particle because it's made of similar material to living cells. But one of the clinchers is that the virus cannot reproduce by itself. And so this is kind of our conversation, is that the only way it can replicate and make more of itself is by infecting what's called a host cell. In this case, it's going to be a bacteria. And so let's look at the structure of a virus because it's important. And so it has this outer coat made up of protein. And so if I were to draw that three-dimensionally, it would look more like a polyhedral. It had many sides to it, sort of like that structure that you find at Spaceship in the Epcot Center, if you've ever been to that down in Florida. But it's got this protein coat, or this head area, and it's got this body right here that's sort of like a straw, a hollow straw, and it has these tail fibers made up of protein. So it's got this protein, and like, what's inside of it? Inside of it, it could either be, let me go blue, could either be DNA or it could be RNA. And so different viruses have different genetic material. And so let's just stay with DNA as being inside. And so this is your basic structure of a virus. And you're like, well, how does it work? Well, basically, it needs to infect a host cell. And so this is what we want to look at in our conversation. And so evidence from that DNA was the genetic material comes from understanding how bacteria that get infected by these viruses. And so let's take a look. And as I mentioned before, viruses contain either DNA, sometimes RNA, and they're enclosed in this protein coat, and that's significant. And so this is a really cool picture. This shows right here, let me illustrate this. This is showing, if I can, a bacteria. And so this is a bacteria, and the bacteria is in bad shape. It's kind of busting open, kind of like a piñata would bust open with candy, if you remember these days of a baseball party when you swing a bat and bust open a piñata and all the candies come out. In this case, these are viruses. And so what a virus does is it comes down like this and it lands on the outside of a bacterial cell. And so they land on the outside. And if you think of the protein as being sort of like a syringe, from our previous conversation, and so what the protein is going to do is stay on the outside, and the virus that is going to inject its DNA inside the bacteria. Now I'm going to put that for vDNA. Now what's brutal or insidious about this is that the bacteria has its own DNA. And what does the DNA code for? If you recall this from previous discussions, is that it's the recipe to make proteins. And so this is what the bacteria does. It makes bacterial proteins, it grows, and then it gets to a point, and then it divides. But what's brutal about this is that when a virus injects its DNA into the bacteria doesn't know the difference between its own bacterial DNA and the viral DNA. And so in this case, what it will do is it'll start reading the viral DNA as its own. And what will it do? Well, that will produce viral messenger RNA, which will then go to the bacterial ribosomes. And so this is the key. Bacteria, which are living things, they have ribosomes, which you remember as the site of protein synthesis. And so the bacteria ribosomes, when they read the viral RNA, they will start making viral proteins. And these viral proteins, sort of like the little candies inside, will build up, will build up, will build up. And how does it do that? Well, it uses the bacteria's amino acids to make more viral proteins. Isn't that brutal? And then it even uses, this is even more brutal, it makes more copies of viral DNA, and it places this inside. And then eventually the bacteria becomes so engorged with viruses that it actually lyses or ruptures or busts open. And that's what you see here, is all of these viruses which are going to go out and start the infection of other bacteria. And so it's kind of like a little bit of a hijacking. The virus comes in, it sort of, injects its own DNA into your car, if you will, if it's a car jack, and basically it dices up. I'm not going into the detail, but I could. It dices up the bacterial DNA, and it sort of commandeers the cell and turns it into a virus factory. And so okay, what's that? All that have to do with DNA being the genetic material? And so it's an interesting story, because here a couple terms that we might be needing is that a virus is sometimes also called a phage, and it's a Greek word meaning to eat. And so it isn't literal like where the virus is eating the bacteria, but these are called bacteriophages, which are particular. Viruses are very specific. In other words, if we got bacteriophages, or viruses that infect bacteria, into our body, they wouldn't hurt us, because we would only get sick by animal viruses, and plants only get sick by plant viruses. And so bacteria get sick by bacteria viruses called bacteriophages. So these bacteriophages led us to the understanding that DNA is the genetic material. Now, how is that? Well, again, this is a beautiful picture showing a bacteria. Now, I'm starting to feel sorry for the bacteria, because you can see what's happening. The virus, or the protein, should I say, stays on the outside, and what you're not seeing, but I will draw, is that the virus is injecting its viral DNA inside. And by injecting the viral DNA inside, it's able to take over the cell, and it produces many viruses inside, and eventually the cell will rupture, and then all these viruses will get out. So the key is, let me illustrate this. The key is, let me stick with this color red for a second. The protein stays on the outside, and the DNA, which I'll make blue, is able to go inside the bacteria. And you're like, yes, I understand that, but what's the point? Well, if the question of the genetic material, is the genetic material, remember this is the point, is the genetic material protein or is it DNA? It's like, which is it? And you're like, well, I know that it's DNA. Well, yes, so do I. But how did the experiment show that it was DNA that that causes bacteria to die and the protein did not. Well, if you had this picture back in the 1950s when this experiment was conducted, there'd be no need for the experiment because you can clearly see the protein coats are on the outside and the DNA is able to go inside. But they did not have this scanning electron microscope back in the 1950s. And so Hershey and Chase were rather clever. They used very simple apparatus and equipment. They used like a blender and a centrifuge and some radioactivity to determine this. And so let's get into this for a second. So how can they show, for example, this is what's happening, but it's microscopic. You cannot see this. So what do you have? What can you see? Okay, so what can you see is you can have a test tube like that. Let's draw that in, a test tube. And let's put some liquid in here. And let's draw the bacteria. Let's go here, red. Let's draw the bacteria in here like this. So they're floating in here. You're growing bacteria. And then the viruses are in here as well. And so the question is, here are the little viruses on the outside, the protein part. And it's like, well, how are you going to show that the protein are on the outside and the DNA went on the inside? Well, what they did was they knew a little bit about the structure of a protein, Hershey and Chase did. And they knew that proteins, like you do, are made up of chains of amino acids. And these are each amino acids. And the difference between one amino acid and the next are these little R groups that come off like this. And there's 20 different R groups. And so my point I want to make is one of the R groups has sulfur. I'll just put S as its side group or its side chain. And so what Hershey and Chase were able to do is when they were growing this medium here of bacteria and viruses, they were able to use radioactive, I'm going to put like an asterisk there, they were able to use radioactive sulfur in the growth medium. And in so doing, all protein will contain radioactive isotope sulfur, and therefore this can be detected. The protein can be detected using radioactive sulfur. This is my point. Now, no one at this time knew the structure, the complete structure of DNA. We now know the complete structure of DNA as being the double helix. And that's coming in our next discussion, the structure of DNA. But at this time, when Hershey and Chase performed this experiment, the only thing they really knew so much with confidence about DNA, let's see if I can draw this. And so DNA is on the inside. What they knew about DNA was that DNA had in its components the element phosphorus. Now, it's like, of course, protein has carbon, of course it has oxygen, of course it has hydrogen, so does DNA has carbon, hydrogen, and oxygen. But what's unique about DNA is that it had the element phosphorus in it, and what's unique about protein is that it had the element sulfur. So they grew, again, the bacteria and viruses in a medium that contained radioactive phosphorus. And so the DNA was labeled with radioactive phosphorus and the protein was labeled with radioactive sulfur. And so now they're going to be able to use this to determine something. And so let's take a look. And so again, actually I thought you might want to see what these guys look like, and actually it's a woman here. This is Martha Chase and this is Alfred Hershey. And again, this is early 1950s. And again, this is a great picture showing a bacteriophage. Here's the protein on the outside, and here's the DNA being injected into the bacteria cell. If Hershey and Chase had this picture, they wouldn't need to conduct the experiment that they did. They wouldn't have to have labeled the radioactive protein and the radioactive DNA because you can clearly see the protein stays on the outside. And so again, this is their experiment. So in 1952, Hershey and Chase showed that DNA was a genetic material, and it was a landmark experiment because it was now definitive that DNA was the genetic material. And so how did they do it? And again, I mentioned that it was rather simple. They simply used glasses and flasks, and they grew. Let's look at this top part first. And they grew inside here. Now again, you cannot see this. So it's microscopic. It looks like just a clear liquid. But they used radioactive sulfur, which is isotope 35. And as you can see here, the protein, let's go pink, the protein is going to be pink. And the cells are not going to have any pink in them because the protein, remember, stays on the outside. And so do you see how the bacteria are clinging to the outside? And so what they did is they took some of this liquid and put it into a blender. Now, they didn't hit liquefy or mix at the highest level, but they just sort of spun it just briefly, spin, spin, spin. And when you spin it just briefly in the blender, what happens is you dislodge the phages from the outside. In other words, they go flying off. And so this is in the liquid. And it's like, well, it's all liquid. Well, as it turns out, you can take this liquid and put it into a test tube. And in the lab, they have a centrifuge. And a centrifuge basically is a machine where you place these test tubes in, and it rotates the test tubes very quickly. It spins them around and around very, very rapidly. Kind of reminds me of like a ride at Great America where you're spinning around. And as it turns out, the most dense objects in the liquid, now this is sort of like, if you think of this as Italian salad dressing, do you know like the spices, when you set it on the table for a long period of time, the spices sink to the bottom and the oil is on the top? The oil on the top, or the liquid, is just the food that they were growing in, called the supernatant. But the cells themselves are heavy, and the cells pellet to the bottom. And so as it turns out, when you look at this, you can see that the cells on the bottom down here do not contain any radioactive sulfur. All the radioactive sulfur is here in the liquid, or in the oil, if you will, which showed that the radioactivity in the protein coats, did not enter the cell. So the protein coats were on the outside. Again, we already knew that from what we were talking about before, but Hershey and Chase did not know it until they saw that the sulfur, therefore the protein, did not enter the cell. Now check this out down below. When you grow the same phages and bacteria in a medium that contains radioactive phosphorus, you can see DNA is now labeled blue for radioactivity. And as we said before, the DNA gets injected into the bacteria. When it's injected into the bacteria, that's what causes the ribosomes to produce more proteins. It causes viruses to replicate, and therefore causes the cell to puncture open. So it's the DNA that's doing all the work because it's going inside the cell. How do you show that? Well, again, you can't see anything. It looks like just a liquid, but again, it's the same thing that's happening up above. But in this case, we put it in the blender, spin, spin, spin. Lo and behold, the proteins go flying off. But this time we didn't label the proteins. This time we labeled the DNA. And in this case, since the DNA went into the cell, that means the cells will be glowing blue because the DNA is inside. So check it out. So when you put it in the centrifuge and the liquid goes in the tube, spin, 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 all the heavy cells go to the bottom, and it's the DNA that's inside the cell. Thus, it's DNA that went inside the cell. It's DNA that's the genetic material that causes viral replication inside of bacterial cells. It's rather important. And so at this point, the whole biological community, the whole world is now convinced that DNA is the genetic material. But it's like one of those things where in life, when something's determined definitively, like DNA is the genetic material, then everybody starts coming out. Like when the Giants won the World Series. It's like, I knew the Giants were the best team. I knew they were going to win the World Series. Once DNA was determined to be the genetic material, then other scientists started coming out saying, well, I knew it the whole time, because there was some circumstantial evidence that showed the DNA could have been the genetic material. I just want to talk about that briefly. Scientists knew at around the turn of the century in the 1900s that they didn't have this great of a picture, but they knew that the cell, as you now know, produce chromosomes during prophase of mitosis before they divide. And so here's a chromosome. They were able to pull chromosomes or extract chromosomes from cells. And you're like, well, duh, then they will know that DNA is the genetic material. Not so fast. Because, as you know, the chromosome is DNA wrapped around proteins. And so it wasn't very clear exactly that DNA was the genetic material. It could have been protein. However, now that we were very confident that DNA was a genetic material, we were then thinking, it is true that the cell does partition chromosomes right before the cell divides. Chromosomes probably are the genetic material in some way. And then it became clear that the amount of DNA that was contained inside the cell was doubled prior to mitosis. And you know that the DNA replicates itself during interphase. And that if you can take a look at the amount of DNA once the cell divides, it's then half the amount during interphase after S. And so it became clear at this time that DNA was a genetic material. And so at this point, it was a race because scientists knew that whoever was able to determine the structure of DNA would be one of the most famous biologists of all time. So I hope you enjoyed this discussion of the Hershey-Chase experiment. Thank you for watching.